In this presentation, I'll introduce my latest publication, Motorless Transport of Microtubules along Tubulin-RanGTP and Salt Gradients. It was published in Nature Communications on November 1st, 2024. The main idea of this work is about identifying diffusiophoretic transport of stable microtubules along cytoplasmic and higher concentration gradients of soluble tubulin, RanGTP, and magnesium salts. The motorless motion of intracellular macromolecules along various concentration gradients of smaller species is a popular research question in biology and biophysics. Such a transport phenomenon is called diffusiophoresis in colloid science. We focus on the fundamental aspect of diffusiophoresis as a physico-chemical hydrodynamic phenomenon and take a step-by-step approach to validate the observed behavior of microtubules as diffusiophoresis. Now let's look into the details. Microtubules are dynamic filaments that form spindles in eukaryotic cells during mitosis and meiosis. It has a cylindrical shape with a 25 nanometer outer diameter, and its length can vary up to tens of micrometers depending on the intracellular location and the stages of cell cycles. Among many species present in cytoplasm, it is well known that the RanGTP gradient is a critical regulator of spindle assembly and positioning in both mitosis and meiosis. Also, it was recently visualized that there is pre-accumulation of tubulin molecules near the nucleation sites of spindles. Given the concentration gradients of relevant species and the transport of microtubule segments in spindles, we asked if diffusiophoresis of microtubules can be present during spindle assembly. Diffusiophoresis is the spontaneous motion of particles along a concentration gradient of solutes. Validating the occurrence of diffusiophoresis in spindles is not as simple as it sounds. Diffusiophoresis is a physico-chemical hydrodynamic phenomenon driven by the osmotic flow generated along the particle surface within nanometers of interaction length scale. If a negatively charged particle is put under a simple salt gradient, diffusioosmotic flow is generated along the particle surface. As the particle is mobile, the shear stress will make it move to the opposite direction, and this motion is called diffusiophoresis. Therefore, when analyzing the particle transport in such a configuration, it is necessary to identify the coupled interactions among particles, solutes, and the fluid flow. All experiments in this project were conducted in a microfluidic channel with a dead-end pore geometry. By using different initial and boundary conditions, we either entrained microtubules into initially empty pores or compacted existing microtubules along various concentration gradients of solutes. In this way, we can set up a one-dimensional concentration gradient of solutes and observe the response of stable microtubules that are approximately 10 micrometers long. Among many solutes that potentially influence the behavior of microtubules, we thought the most important one to investigate is the soluble tubulin. Inside a cell, microtubule generation requires that the concentration of tubulin near nucleation sites be above a critical level. Therefore, near microtubule structures, it is natural that a concentration gradient of soluble tubulin is present. We used two different buffers as surrounding media and observed entrainment of stable microtubules down the gradients of micromolar tubulin. At this point, we cannot yet state that the motion of microtubules is diffusiophoresis, but we notice a few important features from this set of experiments. First, the entrainment distance of microtubules depends on the tubulin concentration gradients. Second, the buffer type affects the microtubule transport. Lastly, the entrainment fronts in all experiments have a parabolic shape, meaning that there can be the influence of the slip flow generated along the wall in the presence of the tubulin gradients. Before analyzing the phoretic behavior of microtubules, we must rule out the effect of liquid flow generated inside the pore. The parabolic entrainment front indicates that the imposed tubulin concentration gradient induces osmotic flow along the pore walls, making the flow velocity inside the pore non-uniform. The rescaled flow velocity can be plotted versus distance across the pore, and we can detect a y position where the flow velocity becomes 0. This means that if we track the microtubule entrainment front along y equals 40 micrometers at each time point, we are detecting purely the phoretic component of the apparent microtubule motion. The graphs show clear concentration dependence of microtubule entrainment. Since we only visualize the effect of tubulin concentration gradient on the apparent travel distance of microtubules, we must further identify all the interactions listed here to fully validate the diffusiophoretic mechanism underlying the observed microtubule migration. We take a step-by-step approach to separately identify the interactions between microtubules and background species, interaction between tubulin and background species, and finally, the interaction between microtubules and tubulin molecules in the presence of the background species. We next conducted 2 sets of experiments using magnesium chloride gradients in BRB80 buffer. In the first set, we observed the response of microtubules under magnesium chloride gradients, and in the second set, we observed the behavior of commercial polystyrene microspheres. Both the microtubules and polystyrene particles move up the gradient of magnesium chloride. The behavior of polystyrene particles is straightforward as it can be directly analyzed with an electrolyte-driven diffusiophoresis and diffusioosmosis model. Therefore, the configurational analogy between the 2 sets of experiments can tell us more about the magnesium chloride-driven migration of microtubules. We plotted the entrainment distances of microtubules measured along y equals 40 micrometers for 6 different magnesium chloride concentration gradients. As shown in the linear plots, the entrainment behavior of microtubules indicates diffusive nature of this transport process. For the magnesium chloride experiments, we analyzed 2-dimensional entrainment profiles by detecting the particle front at 5 different y positions. In all experiments, microtubules migrated fastest along the center line and slowest near the wall, making the front shape parabolic. To validate a diffusiophoretic mechanism for the microtubule behaviors along magnesium chloride gradients, we used a multi-ion diffusiophoresis and diffusioosmosis model. The model considers coupled transport of all electrolytes in the liquid phase and the diffusioosmotically induced liquid flow inside the pores. Particle trajectories along different y positions were obtained by considering local flow velocities at the corresponding y positions. The measured and calculated trajectories are compared directly by using the zeta potentials of the microtubules and the pore walls as fitting parameters. For 25 millimolar magnesium chloride, we obtained the best fit with the wall and microtubule zeta potentials of -7.2 and -35.5 millivolts respectively, which are consistent with the previously reported zeta potential values. Therefore, with this comparison, we confirm that the magnesium chloride gradient-driven migration of microtubule is diffusiophoresis. Comparison between the model and experiment was done for all magnesium chloride concentrations, and we conclude that microtubules can undergo diffusiophoresis in the presence of magnesium chloride gradients in BRB80 buffer. The main parameter for the surface interaction between the ions and microtubules here is the negative zeta potential of microtubules. We also confirmed that magnesium chloride-driven diffusiophoresis of microtubules and polystyrene particles occurs similarly in the CSFXB buffer with different magnitudes of the fitting parameters and the entrainment distances. We now move on to the next part, identifying the interactions between tubulin and background species. We examined the diffusion behavior of soluble tubulin separately using 5 different background solutions without any microtubules present. We observed that tubulin diffusion is largely unaffected by the background species, and the measured diffusivity of soluble tubulin is approximately 10 to the minus 10 meters square per second, consistent with the known size of the alpha-beta tubulin dimer. The results suggest that when analyzing the tubulin gradient-driven migration of microtubules, we're allowed to consider only the diffusion of tubulin without accounting for the coupled transport of the background species. Finally, we're almost set to better understand the details about tubulin-driven transport of microtubules. Before returning to the microtubule results, we conducted a set of experiments with polystyrene particles under micromolar gradients of soluble tubulin. Analogous to the microtubules, polystyrene particles migrated down the tubulin gradients. We applied the non-electrolyte diffusiophoresis model to analyze tubulin-driven migration of polystyrene particles, and compared the model and experiments by setting the interaction length scale as the fitting parameter. The 100 nanometer interaction length scale was larger at lower tubulin concentrations, indicating that tubulin molecules are more widely spaced under these conditions. We considered the repulsive interaction between tubulin and polystyrene particles, which is consistent with the negative surface charges of both. Since the analogy between microtubules and polystyrene particles holds due to their negative surface potentials and micrometer size, we can apply the same non-electrolyte diffusiophoresis model to the microtubule experiments and examine whether the interaction length scale is also consistent. The obtained values for the interaction length scales in the microtubule experiments were also around the 100 nanometers. The scale and trend matched the average separation distance in micromolar tubulin solutions, making our step-by-step validation and conclusion about microtubule diffusiophoresis physically reasonable. We suggest that there is a long-range interaction between microtubules and soluble tubulin due to their negative surface potentials, and that this interaction is weaker in BRB80 buffer compared to CSFXB, resulting in a smaller diffusiophoretic travel distance. Our surface potential argument as the main source of the long-range interaction is further validated using amine-modified polystyrene particles which have a positive surface potential. We confirmed that the amine-modified polystyrene particles migrate up the tubulin gradient, opposite to the migration direction of microtubules and carboxylate-modified polystyrene particles. So far, we have identified the diffusiophoresis mechanism for microtubules in the presence of tubulin and magnesium chloride gradients within buffer environments. In the next part, we present experiments with other solutes and medium to show that diffusiophoresis of microtubules can be present in the cytoplasmic environment as a motorless transport mechanism. Another important protein gradient that arises during spindle assembly in addition to tubulin is RanGTP. It is known that the RanGTP gradient is a critical regulator of spindle assembly and positioning, so it is necessary that we identify possibility of RanGTP-driven diffusiophoresis of microtubules. We first tested both directions of RanGTP concentration gradients and observed the response of microtubules. Unlike the previous experiments, it appeared that the microtubules move both up and down the gradients of RanGTP in CSFXB buffer, with little concentration dependence and smaller travel distances. To understand this two-direction behavior of RanGTP-driven transport, we investigated further details of the entrainment setup and observed that there is depolymerized tubulin with fluorescent signals diffusing in the background. Therefore, we investigated the cross-diffusion behavior of tubulin and RanGTP separately and found out that the tubulin diffusion was enhanced at higher RanGTP concentration gradients. It must be that in the microtubule experiments with RanGTP, the generation and diffusion of the depolymerized tubulin is never negligible and is always affecting the diffusiophoretic behavior of microtubules. To rule out the microtubule depolymerization, we use polystyrene particles and examine the directionality of RanGTP-driven diffusiophoresis of negatively charged particles. Without any background tubulin, the negatively charged particles always migrated down the gradients of RanGTP, and only in the presence of the cross-diffusion of tubulin and RanGTP in the background, the particles migrated up the RanGTP gradient. This is why the microtubule diffusiophoresis show the two-direction behavior and little concentration dependence along the RanGTP gradients. In the published work, we also included the model predictions for the RanGTP-driven diffusiophoresis of microtubules, and we concluded that microtubules can undergo diffusiophoresis along various relevant solutes of spindle assembly. Finally, we reproduced the diffusiophoresis of negatively charged particles in an active cytoplasmic environment using the Xenobus-Lewis egg extract. Polystyrene microsphere was chosen as a representative negatively charged particle to rule out other physiological processes that could occur with microtubules. We confirmed that magnesium chloride, tubulin, and RanGTP-driven diffusiophoresis occur even in the frog egg extract condition, which lead to our final conclusion that microtubule diffusiophoresis can be present during spindle assembly. More details of this work can be found in the published article. Thank you for watching this video, and please let me know if you have any questions.